we are perfectly on time. Now we get to switch gears and talk about a Whitehead Institute program that has stood the test of 30+ years. Founded in 1984, the program emerged from the notion that nurturing young scientists during their most creative years will yield a new generation of leaders more rapidly than traditional programs. And I'd like to say that, you know, for those of you who are entrepreneurs or know of the entrepreneurial world, the key thing here is let people make mistakes early. Whitehead Fellows are given the space, the resources, and the support needed to run their own labs and pursue an independent research agenda. But unlike a traditional faculty appointment, Whitehead Fellows do not do certain things. They do not have teaching responsibilities. They don't have to serve on committees and all sorts of things. They can instead focus exclusively on launching a research program. Is Terry Orweaver here today? I think she will be Terry's going to be joining us at lunch, oversees the process of reviewing applicants to the program. It turns out this is a high-risk, high-reward program. It's evolved, and we want to share the success of some of our fellows. I'll just name a couple here. Peter Kim was one of the first Whitehead Fellows. He joined the faculty here and then went on to become, for a good long run, president of Merck Research Laboratories. He's returned now to the academic sphere. Interestingly, one of our former fellows, Kate Rubins, became a NASA astronaut. She is currently circling us on the International Space Station as we meet. And as Tom mentioned, I was once a fellow, in fact, the first fellow at Whitehead. At age 28, having just completed my MD at Harvard Medical School, I came across here and entered the building the day the doors opened and have never left. Well, I go home sometimes, but there's so much to learn, teach, and to do here. So it's my pleasure to introduce to you this morning two of our fellows who have been deemed so prepared to take their own independent research to the next level and build a lab. And those two individuals are Sebastian Lourido and Sylvie Rouskin. I'm going to ask each of them individually to share a little bit about their research. And actually, Sebastian, are you going first? Do you want to come up? As I begin to introduce this, we'll be trading off some of the technology here. So I'm going to ask each of Sebastian and Sylvie to share something about their research, and then I will have the chance to interview them. And it's a little bit daunting, the prospect of attempting to interview them in Tom Ashbrook's presence, but I will do my best, Tom, right? And then you'll have the opportunity, I'll interview them a little bit, and you'll have the opportunity also to ask them questions. So I think, are we doing the cards again? Okay, so for Sebastian and Sylvie, be writing down your questions on your cards, and we will, and I should say that at our break later this morning and at lunch, you'll also have the chance to speak with Sylvie and Sebastian. So with that, I'll turn it over to you, Sebastian.
Thank you so much. And it's a really tremendous pleasure. This is, at the end of this month, the 4th year that I've been a Whitehead Fellow, and it's wonderful to see so many familiar faces and, of course, so many new ones here at the Colloquium. So this is sort of an unusual photo to get started with, and one that I hope over the course of my talk will bring up many of the themes in basic research, model organisms, and how we as biologists understand the world. Sequoias are amongst the largest organisms on Earth. And we can appreciate their scale here based on the climbers that are trying to get up to the top. They live between 1,200 and 1,800 years. And they are brought up here in the course of my talk so that I can illustrate that we can't study all things in all organisms. Imagine, for instance, trying to study aging in sequoias. It would take our entire lifetime to just notice a 10% change in their lifespan. Or to understand the genetic determinants of height, how long it would take one of those trees to grow. Every so often, however, biologists identify organisms whose specific characteristics allow us to study with precision and rigor particular biological processes. These so-called model organisms act as lenses through which to dissect and understand the biological universe. And many of the laboratories at Whitehead have exploited these particular organisms to inform our own biology. Bacteria, such as Escherichia coli, have been used to understand the molecular basis of biological processes. Yeast have taught us about how these networks of chemical processes serve to explain how cells divide, for instance. And simple organisms like fruit flies have taught us how those cells come together into tissues and entire organisms and develop into such a variety of cell types. Their contributions are absolutely remarkable and have changed the course of biological research forever. But just as with the lenses in a microscope or a telescope, these lenses can sometimes distort or overfocus our attention. If we look at the part of the tree of life, and this is the relationships across evolution of many of the different organisms that belong to a branch called the eukaryotes, to which we belong. That's us up here, along with all of the mammals and all of the animals that we use as molecular systems. And then another system that I just mentioned, the fungi, have served together to highlight a lot of molecular biology. Most of molecular biology has been focused on just a few organisms, and as such, the lessons from those investigations serve to inform only certain branches within this tree of life. Again, we have the situation where the vast majority of the diversity of life is left in utter obscurity. And one of these branches is actually quite important to us and has been one of the fascinations that I've pursued here as a Whitehead Fellow. This branch is called, This branch is that of the apicomplexan parasites, which are a group of about 5,000 species of single-celled organisms that have co-evolved along with the animals that they parasitize over approximately 500 million years. And yet, this unique evolutionary tract is practically unexplored at the molecular level. You might be familiar with some of these organisms because of the infections that they cause. For instance, malaria, which in 2013 caused 200 million infections, many of them leading to the death of children in underprivileged nations. This represents a really tremendous burden on developing countries, and the same populations are susceptible to a second disease that we've recently recognized that will be less familiar to most of you, that of cryptosporidium, which causes nearly a quarter of infant diarrheas, and those diarrheas account for nearly 10% of childhood mortality. Together, these diseases represent a humanitarian crisis that has motivated scientists to investigate the organisms that cause these diseases. My laboratory has taken a different approach. Those organisms are tremendously difficult to culture and investigate, and I commend the many scientists that have devoted their careers to studying them. But in my lab, we study an organism called Toxoplasma gondii, which is quite easy to culture. It too infects nearly a quarter of the world's population. Once infected, individuals are infected for the rest of their life, but in most cases, it's completely asymptomatic. Some of you will be familiar with this disease because of the notion that pregnant women shouldn't change the kitty litter. That's because humans can contract Toxoplasma gondii from cats, and in pregnant women and immunocompromised individuals, the disease can be life-threatening for the life of the fetus or the immunocompromised individual. As I mentioned, in the lab, we can take human cells that we can propagate virtually indefinitely, and we can take these parasites, which are highlighted here, and we can see them actually infect those cells in real time. And once inside those human cells, they can replicate, and we can propagate them, and we can perform all forms of genetic manipulations that allow us to study their biology. So we study Toxoplasma gondii for two reasons. The first one is that undoubtedly it is an important human pathogen for those afflicted. But equally importantly, it can serve as a model organism to study those parasites that are related to it and allow us to investigate the core biology of this group of parasites with unprecedented specificity and efficiency. This is a model of Toxoplasma, and as biologists over the past few decades, we've gotten really good at reading out the genetic sequence from the DNA of these organisms and decoding essentially the 60 million or so bases that constitute the Toxoplasma genome. We can take algorithms and different sort of predictive measures and divide those 60 million letters into functional elements we call genes, of which Toxoplasma has approximately 8,000. Just to highlight the challenge of studying a parasite such as Toxoplasma, as a graduate student, I spent 6 years studying 2 of those 8,000 genes. However, over the past few years, there has been a really tremendous revolution in our ability to manipulate the genomes, not only of human and mouse cells and yeast, but also of rare organisms like Toxoplasma gondii. The basis of this genetic revolution has been the use of CRISPR-Cas9. This is the structure of that tool, and it serves as a pair of molecular scissors that can be targeted specifically to virtually any region of the genome of an organism. The way these molecular scissors, which we call Cas9, are targeted is by instructions that are highlighted here in red, which target a predetermined region of the genome in the form of the DNA. These parasites can be used in a really novel system, which we call a pooled genetic screen, which I'll explain briefly, which can allow us to put a set of instructions that targets each one of those genes, of those 8,000 genes, and create a population of parasites where each one of those parasites is deficient in one of those genes in its genome. We can then culture that population in human cells, and over the course of the experiment, we can see which disruptions are lethal to the parasite, which genes are important, and which ones are dispensable for growth in human cells. We can now do this over the course of a couple of weeks for all 8,000 genes in the parasite genome. This is another way of representing those 8,000 genes as bars in the 14 chromosomes of Toxoplasma gondii. And what we've been able to accomplish is essentially to determine the contribution of each one of these genes to the infectious process, from less important to more important genes, and in particular pinpoint those genes that represent novel vulnerabilities within the Toxoplasma genome that can help us develop new strategies of intervention. This is a tremendous amount of information that will dictate biomedical research in this area for decades to come. But I'd like to just highlight, before I end, one particular example. This is a gene that when we turn off prevents parasites from entering human cells and represents a unique link in the ability of these parasites to invade human cells and to cause disease. We've already demonstrated that this same gene is important for malaria parasites to survive, and we believe that this research will continue to inform the treatment and investigation of these important human diseases. There's another aspect in which I think we need to consider the use of new model organisms, and that's the fact that biological diversity can be an engine for innovation. Anytime we allow scientists' minds loose on a new model organism to investigate and follow their curiosity, we discover new therapies and compounds from plants, new structures and forms of design from marine corals, and because the vast majority of the diversity of life lies within these single-celled organisms and within the microbial world, there's really an untapped resource for human creativity and innovation within this realm of the biological universe. All of this work relies on a really talented team of people and, of course, on the funding that is provided by the Whitehead Institute and the National Institutes of Health, which have allowed us to follow really innovative avenues of exploration. Thank you.
So we're going to hold your questions for Sebastian right now and move on to hearing from Sylvie. And then after her presentation, I'm going to ask both Sebastian and Sylvie to come up here and we'll take questions. Do you want me to get this one out of the way? Here. Sure. Good.
Okay, great.
Sylvie.
Hi, thank you all for coming today. So I'm going to tell you about finding the second dimension of genetic information. Is there a laser pointer that's supposed to work here? It's not what? Oh, it's just an advanced or? Sorry. Oh, okay. So it's this one. Okay, thanks a lot. All right, and so first I'd like to start by defining what is genetic information, as our view has really changed over the years. And the traditional view, I don't know why, sorry about that. Oops, okay. The traditional view, for some reason the laser pointer is not pointing. It's too bad, okay. But the traditional view has been that the genetic information is basically all the information that you need to make an organism. And the traditional view has been that genetic information is stored in the DNA in the form of genes, and then it gets copied to RNA, which then, through a set of instructions that's like a molecular dictionary, converts RNA molecules to protein molecules. And proteins are really those machines that make up the organism. And for a long time, it was really kind of assumed that the genetic information is really just this genetic code that we understand quite well. But in the early 2000s, with the Human Genome Project, with the development of this high-throughput sequencing, we could actually sequence all of the DNA in the organism and ask, well, how much of this genetic information is the genetic code? And when we sequence bacteria, we're pretty happy to know that most of it, here really in green, is the genetic code. So we can understand bacteria pretty well because this is the genetic code that we're very familiar with. But when we sequenced humans, scientists were actually extremely surprised to find out that this genetic code that we know so well is only 2% of the human genetic information. And I think that points to two things. One is that there's a whole lot more to discover. And two, for the purpose of this talk, more important, nearly 50% of this human genetic information codes solely for RNA. So this is this molecule called RNA that actually never gets made into a protein. It just stops right here. So what exactly is RNA? RNA is very similar to DNA, and they're both polymers, meaning they're just chains of building blocks, some small subunits. There are only 4 different types of building blocks, and they're almost exactly the same between RNA and DNA. And when those 4 different blocks get strung together, they make up RNA and DNA. The biggest difference between RNA and DNA is that the building blocks in RNA have an extra oxygen atom. It's too bad I can't point. They have an extra oxygen atom, and really this is where the name comes from. DNA is deoxyribonucleic acid, and RNA is just ribonucleic acid. And this oxygen atom, oxygen, is very reactive and gives RNA very different chemical properties. Oh, is there a laser pointer? Okay, so if you're just all right. So what? Whereas DNA is constrained to one type of shape or structure, DNA molecules, RNA forms many complex structures inside cells. And I like this analogy with a toy that my daughter likes to play with called mag formers, where you can see immediately appreciate the diversity of all those different shapes and how they have completely different properties. But they're all just made from, stringing together 2 types of subunits, just triangles and squares. So very analogously, RNA molecules are just made of 4 different types. And in the linear version, the first dimension, they carry the genetic information. But once they get folded up, like this, you can see how this type of folding serves to specifically conceal parts of the genetic information or expose it. And so this is the main mechanism through which RNA structure or shape will affect gene expression. And those are just different ways that we represent the linear form and the folded RNA structures. So what type of information is encoded in RNA structure? I'm going to tell you just about 2 types of information that we study in the lab. One is this control of localization. So where a gene is copied from the DNA, there are actually multiple copies, so multiple RNA copies of this gene. And initially it was thought that all those copies are just randomly distributed throughout the cell. But this is definitely not true, especially in development. This RNA molecules are very specifically localized. And it turns out that this localization is extremely important in development. And if this doesn't happen properly, it would lead to developmental defects and sterility. Another aspect of RNA structure control is what version of a gene is expressed. And basically, every single gene in humans have multiple different versions. And they're created through this process that in biology is called alternative splicing. But those versions and their abundance is absolutely crucial, especially in the nervous system. And if if the incorrect version is expressed, or the versions are expressed in the wrong ratio, that can lead to dementia and Alzheimer's. Hopefully by now I've convinced you that RNA structure is extremely important. And yet, we know the RNA structure of only a handful of genes, because historically, RNA structure has been extremely difficult to study. So the goals of my lab is really to determine the RNA structure of every gene and decode the rules through which RNA structure controls gene expression, and eventually be able to manipulate RNA structure to prevent and treat relevant disease. So today I'm going to tell you just very briefly about how to determine the RNA structure of every gene. So my lab has been focused on developing new methods to determine RNA structure inside cells. And we use this chemical-based probing. We use a small chemical that enters the cells quickly, and it reacts with the open, exposed surfaces of the RNA structure. And I like this analogy of if we imagine, there just this very simple case, there are two different structures. One of them is kind of folded up like a hairball, and the other one is floppy like a spaghetti. And we use this green chemical to, it reacts with this green chemical. In the hairball, it's only going to react in the surface, whether in the spaghetti, it's going to react all over it. Much like if you throw green dye on a person who is in a crouched position, you would only color his back. But if you throw green dye on a person who's wide open, he's going to get drenched in green. And this is the basic principle that we use, just with a lot more precision, to determine the RNA structures inside cells. So in the past, scientists have spent months determining the structure of a single gene inside a cell. And what we can do now is simultaneously detect hundreds of structures at a single molecular resolution. And developing this powerful approach to look at RNA structure is now allowing us to answer a lot of important biological questions that were not accessible before. And I'm briefly going to tell you, just mention what my lab is working on, three different areas to identify the RNA structure that are important in development. And here's a beautiful image seeing each one of those blue is different RNAs, so you can immediately appreciate how they're very specifically localized. That's extremely important for development, yet we have no idea what are the signals that are causing this localization. And this is what we want to identify. In the collaboration with the Sue Lindquist lab, we are looking at how RNA structure is controlling this expression of different gene versions that is extremely important for neuronal function, memory, and learning. With a collaboration with a lab at Harvard, we're looking at the RNA structure of HIV virus and the structures that are essential for HIV infection, with the hope that by perturbing the structure, we can find a cure for HIV. So with that, I'd like to acknowledge the people in my lab, my collaborators, and especially Andrea and Paul Heafy, which are my donors and has contributed for my position at Whitehead. And thank you all for listening.
Okay, so as I ask Sylvie and Sebastian if they'd like to come up here, and we're getting a third chair. This is great. We can, yeah. If you guys want to come take seats and pass your questions along if you've got them. I see some cards are popping up. We'll have time for a few questions here. So, okay, you guys are set. Just grab some seats. So let me ask, so first of all, I should say that in introducing you, I very rudely did not introduce you, right? I didn't say a word about But I think it would be fun to hear, it turns out the Fellows Program has, over the years, brought people from a lot of different backgrounds and a lot of different parts of the world. So where were you guys born, and what was the path by which you sort of ended up here? Who wants to, Sebastian, you want to go first?
Started.
Yeah.
So I've had a little bit of a checkered past. I don't know.
This is why I didn't want to introduce him.
Exactly.
Right.
So I was born in Cali, Colombia, in South America, and my mother was a physician and a medical geneticist. And so I think that early on really inspired a lot of curiosity around sort of the molecular basis of biology. And finishing my high school degree in Colombia, I did my undergraduate at Tulane University in New Orleans. And then I was sort of curious about how science was done elsewhere, and I moved and worked in Berlin at the Max Planck Institute for Infection Biology for a couple of years, sort of as a technician embedded in one of the labs there, after which I went to do my graduate studies in Washington University in St. Louis, which is where a lot of the curiosity around apicomplex and parasites started. And then in 2012, I moved to Whitehead to start my independent laboratory as a Whitehead Fellow.
Sylvie.
Hi, I'm not sure if my mic is working, but hopefully you guys can hear me.
Think you can hear?
Yeah.
Good.
Okay,
Good.
great. So I was born in Bulgaria. My parents were rock stars. They were very loud.
I don't know if you heard that. Sylvie's parents were rock musicians.
They played mostly in Europe. They have a rock band, but I was never interested in music because they were always rehearsing and it was way too loud. And
typical story, right, of kids complaining of their parents making noise, right?
That's right. And when I turned 15, I had this opportunity. I've heard about this exchange program where you can go to the States and get an education. And so I signed up for it, and I left Bulgaria. So I went to Idaho, and.
Makes sense.
And it was a little bit of a culture clash, so I basically after that I went right to college because for multiple reasons, and I thought that I was ready, and so I applied and was able to go to college to Florida. Which was very, very international, and I got my degree in physics because it was really mostly because my physics friends said that, oh, you're just doing biology because that's easier and never do physics. I got pretty upset. I was always a biochemist, but I said, well, I can do physics if I wanted to, so I switched my major and just did physics. But then, of course, I went back to biology to do my, first I was a technician actually in UCSF. I was really interested in viruses. I've always been interested in genetics and genomics, and I was really lucky. I was among the best scientists of the world at UCSF, and it was really fantastic. And I stayed there for grad school, and after that, I had this amazing opportunity to come to the Whitehead. So here I am now.
Wow. You see, everybody has a perfectly straight path. It was obvious how each of these individuals would end up here as members of our Whitehead Fellows program. I want to ask, how has, so being a fellow, the alternative might have been to become a postdoc, right? That would be the standard thing. You'd go on to a next training phase in a lab where somebody else is more or less calling the shots. How has being a Whitehead Fellow, what's the experience of being a Whitehead Fellow been for you, as a sort of thinking about the alternatives that, or free associate?
This is a little bit more recent for you. Do you want to?
Sylvia, you want to pick it up?
All right. Well, I love calling the shots. No, but it's been really an amazing experience for me, because I'm finally able to really follow my curiosity and be able to do what I think is important and get people excited about it. It's been absolutely great. And two of the people that I'm working with, which are just an incredible set of people, just being here at the Whitehead, there's so many incredible people to work with, both to collaborate with or having to join your lab. And I've been just extremely happy and grateful to be able to do this.
You want to add anything, Sebastian?
Yeah, I mean, in a way, the Whitehead is, having been at the Whitehead for as many years as I have, is a great backdrop to asking that question, in part because we have some phenomenal postdocs and phenomenally successful postdocs, right, who've started companies, who've gone on to careers in academia and so on. And I think I now appreciate much more what a postdoc can offer in terms of a scientific career, and that is a tremendous amount of mentorship, oftentimes the ability to switch directions in research, to learn new techniques, and to have almost like a longer incubation period. That contrasts with this sort of immediate drive to follow your own curiosity, oftentimes ill-equipped and dependent on the kindness of strangers, as my neighbor Bob Weinberg, who's often answering questions, and certainly my other colleagues at Whitehead, who help sort of provide the right environment to be able to launch oneself into this very risky pursuit.
Sebastian, a follow-up question for you. You arrived here at Whitehead about 4 years ago, or maybe a little less.
Almost 4, yeah.
Almost 4 years ago. You described some work. You described some very exciting recent work to us. Were you planning to do that work when you arrived?
Yeah, not at all. I mean, if we go back, which I sometimes do, to what I originally proposed to do.
Go all the way 4 years back, yeah.
But no, like what I proposed in that document when I was applying for the Whitehead position, there are many elements that we've carried on, and I think, I won't say we haven't done anything on that, especially in front of the director. But
it's all right. All is forgiven. That's right.
But certainly that flexibility to pursue new directions, which emerged from technologies that were being developed by other labs at the Whitehead, and our ability to immediately apply those technologies, often before they had even been published, to a brand new biological system, a brand new question, and really open up parasite genetics in a way that is unprecedented. Those opportunities were crafted and emerged from both the financial flexibility that the Whitehead Fellows Program presents, and from the intellectual flexibility that the position encourages.
All right, I guess I should have asked that question privately, but felt good to do it here in public. So, Sylvie, here's a question that comes in from our audience. Do environmental inputs influence RNA structure and thus gene expression? And you could interpret that as you will. The questioner asked about diet or exercise or stress or sleep, but you could think about any environmental circumstance.
This is really an excellent question, and it's actually a pretty hot field of research right now. And the way we look at this right now. We're at the state where we're really looking at what's happening relative to the cell, not really relative to the organism. But we do take the cell and we stress it. You know, we in various ways. We shock it with put it in the cold or heat it up, and yes, it does influence RNA structure and gene expression. And you know, we still have a lot more to learn, but I think you know, it would be very exciting, of course, to be able to do that in an organism context and figure out how that translates into the level of the organism and how that would. But there is a lot of evidence now that yes, it does change the RNA structure, and yes, by that, it most likely would also affect gene expression through that.
Great. Sebastian, a question from the audience. You talked about a protein that when mutated or deleted affected internalization in mammalian cells. Are you looking for compounds that would inhibit that protein?
Absolutely. So one of the reasons we zeroed in on that particular protein was in part because it's present in every single genome of this family of parasites. And so it represents actually a unique amongst the proteins that we've identified, a unique conserved aspect of that invasive process. And as such, I think one of the hopes of research over the next couple of years is to identify means of disrupting its function, either through compounds that prevent the interactions that it's performing in order to mediate internalization, or through genetic means of introducing kind of other perturbations, or in fact using it as a new vaccine candidate. This gets a little bit into the weeds, but traditionally, at least in parasitology, a lot of the vaccine candidates that have been explored in terms of malarial vaccines emerged from just looking at the most abundant proteins on the parasite. And so they weren't really focused on the most important proteins, just the most abundant proteins. And that sort of disconnect between function and antigenicity is something that we're interested in exploring, and perhaps going sort of backwards, looking at what are the most important elements and seeing whether those are better vaccine candidates than the really mixed results that we've gotten with the ones we've selected thus far.
Okay. Tom, how'd I do? Is it okay?
Great.
All right.
Thank you very much. Thank you. Thank you. Sylvie and Sebastian, thank you so much. That was fantastic. Thank you. Thank you, David and Sebastian and Sylvie, for that insightful look into the Fellows Program. I mean, when I don't know about you, but I find this incredibly exciting. When I saw that map that you showed, Sebastian, of Africa with that incredible huge density of malaria right in the middle of that continent, and what a drag that has been forever on the human development of this enormous population, solve that, and you could have a very different face of life and development and health and exuberance in Africa. And Sylvia, the idea that we don't know RNA structure, it's like, it's another one of these things was like, why not? How can we possibly, I mean, that's another Dark Ages thing. Let's get out of those Dark Ages, and that you're doing it. I love the thrill of discovery that we're seeing here. I'm reminded again of how much individuals with these incredible backstories matter, whether it's in research or in our lives. I mean, who knows whose life in this room or beyond may be saved by research like this one day. And individuals, each individual who's saved really matters. Think about that as you think about your support for Whitehead. Now, a few words from John McCormick, who is a member of the New York leadership team for the Board of Associates. By day, he is managing director of Health Point Capital in Manhattan. John.
Thank you, Tom. Thank you, Tom, and thank you, David. This has been an outstanding day, and I'm looking forward to the rest of it. Before we move on to a break, I wanted to say a few words about what it means for me to be involved with Whitehead. As an MIT graduate and as a partner in a medical technology investment firm, over the past several years, I have learned things from Whitehead that I could have never learned otherwise. Attending events like this in Boston and in New York, speaking with Whitehead scientists, downloading their papers off the web, and also reading Whitehead publications makes an ongoing education in cutting-edge biology available to all of us. And I never had to take a final exam.
That will change.
It is nonetheless an honor to be associated with this outstanding institution. We Whitehead enthusiasts in New York, when we're not thinking about our sports rivalries with our Boston friends, are working together to spread the news about the Institute beyond Cambridge. So yes, please do tell your friends. Yes, please do tell your colleagues. Yes, do please tell your neighbors about this special institute. Think of yourselves as ambassadors. If every single person in this room can encourage another to get involved, we will have a continued strong showing at future events and give the Board of Associates its already known impact ratio a multiplier effect. I'll bet many of you here learned about Whitehead from a friend. I know I was first introduced to Whitehead by a friend, David Mitchell. Many of you don't know David in this room, and I'm so grateful to David for introducing me to Whitehead, and I'm so grateful to Whitehead for the incredible science that happens here every day. My way of giving back was accepting an executive committee role for the Board of Associates in New York and making introductions to businesses and friends who likewise became involved. You can give back meaningfully to Whitehead even just by encouraging just one friend to get involved. So, I invite many of you who have questions to speak with me at any point later in the day, or let's be in touch after the event. My deep thanks to each of you on the Board of Associates for all you do throughout the year to help advance the Whitehead science. If you are interested in joining the Board of Associates or learning more about the Board of Associates, please see Liz Callanan here, or myself, or any of the other members of the Board of Associates here today. Thank you.
Thank you.